So I will introduce you in Spanish. I always do that in Spanish.
It probably sounds way better.
Okay, and okay, well, bueno, buenos días a todos. Gracias por unirse una vez más con nosotros en nuestro penúltimo seminario del semestre en Frontiers in Genomics. El día de hoy tenemos el gusto de tener con nosotros al doctor Charlie Boone. Él es profesor del Departamento de Genética Molecular de la Universidad de Toronto y del Donnelly Center for Cellular and Molecular Research. Y después de obtener su doctorado en la Universidad de McGill en Montreal, hizo después un postdoctorado en la Universidad de Oregon y después de eso empezó su grupo independiente y siempre trabajando en el modelo de levadura. Su trabajo le ha llevado a ser reconocido como realmente un líder en el campo de la genética de las levaduras y ha recibido varias distinciones, entre las cuales es Fellow de la Royal Society de Canadá, ha recibido varios premios a nivel internacional y es un International Scholar del Howard Hughes Medical Institute. Su investigación está enfocada en establecer un modelo funcional de una célula utilizando el modelo de levadura y para esto han desarrollado una metodología compleja que les ha permitido analizar cientos de mutantes en distintas combinaciones para finalmente establecer un modelo de cómo los diferentes genes interaccionan entre ellos para regular las funciones celulares. Y con esto, pues nada más, pues le quisiera agradecer una vez más al doctor Boone su presencia con nosotros, aunque sea de manera remota. And Charlie, thank you so much for being with us. And with this being said, and I said nothing wrong about you, promise.
Okay.
And I'll leave you now.
Okay. All right, well, thank you very much. And I'll start sharing my screen here. And just let me know if this looks good.
It looks good.
Okay. And you can still see it?
Yeah.
Excellent. All right. Well, thanks so much for inviting me in the introduction. It's, I feel like I'm traveling somewhere, even though I'm not. It's great to see other scientists from around the world. And so today I am going to try and take you from yeast to human cells with these genetic interaction networks that we're mapping. And please feel free to just chime in and ask questions as we go along. Since it's a Zoom talk, that's going to be easy to do. And I would very much welcome that. Okay, so this looks, you can see this, right? So just to give you some perspective, here's, you guys know where you are, way down here, and then we have these, you know, these other humans in between us. But there's Toronto right there. I hope, Yvonne, you can see my pointer like yesterday, right?
Yes.
Okay.
Yes.
Perfect. And you can see how far south we are. Like we're basically Northern California, Southern Oregon, and people think we're way up in the north because it is in fact cold here. Although we do have this, you know, Great Lake effect. And if you don't know much about Canada, everybody lives along this border, basically. You know, it might go up to here. Up in this whole area, which probably is, you know, close to the size of Mexico. I don't know, but probably there's only 50,000 people and there's no roads or anything up there. So it's a wonderful place to explore because you can, there are a few cities, like there's Yellowknife is somewhere around here and you can fly there and then charter your own plane and do all kinds of stuff. But being down here, this is the view out my window. This is the, well, kind of the view out my window. It's the front and the back. But this is the University of Toronto, nestled in the heart of Toronto. Our building, which is, you know, little guy here in this thing is actually right there. That's our building. So we look out over the city, and Toronto has probably doubled in the last 20 years because there's a lot of immigration. So it's turning into a fun place, but it's very much city life. And as I told the students yesterday, our claim to fame at the University of Toronto is that Banting and Best invented insulin here 100 years ago now. This is the centennial anniversary, and Banting and Best discovered it. 1 year later, it was a drug being distributed around the world, and 2 years later, Banting got the Nobel Prize for this. So we haven't done anything since, basically. That's not true, but relatively speaking, it is true. But one thing we're doing intensely here is mapping genetic and chemical genetic interaction networks. I'm probably not going to talk about chemical genetic networks, but you just replace a gene knockout with an inhibitory compound or any type of compound that is bioactive, and you do the same set of genome-wide experiments, and you score chemical genetic interactions rather than genetic interactions, which involve two genes. And we started this with the budding yeast. It's a basic eukaryotic cell with the vacuoles, secretory pathway, mitochondria, and all the goodies like autophagy that go along with that. And many of the genes, several thousand, are conserved from yeast to humans. And so it's been a very tractable genetic organism. It's not as fancy as a Drosophila, but surprisingly, many of the genes that are involved in secretion are in fact playing major roles in driving secretion in things like nerve cells and in flies and humans. Okay, so there were about 1,000 yeast labs around the world, which meant that we had a graduate student for every single yeast gene. And you think it would be easy to figure out how this cell works. It's clearly the most studied single cell in the world at this point. But the community is actually slowly disappearing as people can do more and more things with human cells, but you can never actually replace the incredible genetics that you have with yeast. So there's lots of stuff that remains to be done. We have an incredible database where SGD, where we incorporate all the information about genes. And so we have a pretty good annotation of most of the genes in the genome. It's not as good as you'd like. There are lots of genes, and in some cases, they're tiny little things that are clearly expressed and clearly do something. But even our genetic interaction networks can't uncover their roles. And I'm sure these are littered all over the human genome too. So there's lots of things to figure out, fundamental things that remain to be studied in yeast. But even though it's a relatively small number of genes, there's so many connections between all these genes, and those are what we're trying to figure out with functional genomics. And once we get all those connections and their dynamic qualities in place, then we can think about modeling the cells. So those are the main goals of the yeast community. And one of the most fundamental things you can do is perturb each gene and see what happens. And so this is like, let's figure out the role of each gene. And so this was a project taken on by Ron Davis. Guri Guyver led it, but there are many other labs, Mark Johnson's lab, Mike Snyder's lab. And they basically completed a deletion of every gene in 2002. And you could do this in a diploid, delete one copy of a gene, convert that diploid to a haploid, and then when you do that, we can see that that 20% of the genes, but 1,000 of those genes are essential for cell division. It's a little more tricky than that. You know, some of them are not totally dead. They might divide 50 times and then stop. Some might divide, you know, 1,000 times and then stop. Others, they just sit there as a single spore. So even amongst the essential genes, there's sort of a gradient of phenotypes that still need to be explored. But at first glance, 1,000 genes, 20% of these genes are essential. The other guys are largely non-essential and almost have no fitness defect. There's probably, of the 5,000 viable deletion mutants, there's probably 1,000 that have a pretty severe fitness defect. And one could argue that they should be grouped with the essential genes. But there's a lot of them that really don't have any fitness defects in standard laboratory conditions. And so they remain to be studied in a method that went beyond deletion analysis. Wildly, this same type of project was not possible until CRISPR was invented. I mean, shRNAi, you know, kind of knocked down genes, but it was notoriously it had non-specific, you know, it had specificity issues, and it also had efficacy issues. And so you really couldn't get your hand on most of the essential genes until CRISPR came along. And then several labs did independent studies and defined in a couple different cell culture systems, they just defined the core set of essential genes, which, like in yeast, is about 10 to 20% of the genes that are expressed in a cell line. And the cell lines only express about half of the human genes in general, but they all have to express this core set of essential genes. And so CRISPR really opened that door. So we're interested in this, the idea of the functional connections between all genes. And the way we started assessing this at the University of Toronto, Brenda Andrews and I teamed up to develop this system that we call synthetic genetic array, SGA, where we're looking at the functional connections between genes. And basically what we're doing is making all possible double mutants. And we started with the set of 5,000 viable deletion mutants, but we included, over the years, we made temperature sensitive conditional alleles of essential genes. And these things have been around a long time. They were first invented or studied, I think, in flies, but maybe not. Maybe it was like, some other model organism. But flies definitely had some serious temperature sensitive alleles for years. And yeah, I don't know where they're invented. Someone else can correct me on that if they want to chime in. But anyway, we all know there's some serious genetics going in flies, and you can get TS alleles in flies. And that's irrelevant anyway, because we made all these in yeast. You can do it now with various genome editing strategies. And in fact, we're still making more of them, because it's always a battle to find these special alleles. But once you have one, then you have a system where you can grow yeast at a semi-permissive temperature where they're not dead. So in a TS allele, the yeast cell will grow at a permissive temperature. You shift up the temperature. Usually we think of that as misfolding the protein and revealing the mutant phenotype. And therefore you can play with them at a semi-permissive temperature and do things like score genetic interactions for them. And you couldn't do that with a deletion because the cell would be dead. And so we set out to, do a 6,000 by 6,000 matrix and look at each double mutant twice in 36, million cases. And And so this brings up the idea of genetic interactions, and the David Botstein, who's a brilliant leader of the Yeast community, articulated, the power of genetic interactions to the Yeast community. And Yeast has a single actin allele, ACT1-1. So we write a mutant allele with a lowercase, and then we put a dash on it to indicate the allele. And this allele is TS. The cells are alive at 23. They die at 37. So you can just you can grow the cells up at 23 degrees, plate them out on a Petri dish, and then place that Petri dish at 37 degrees. And if you put, 10 to the 8 or a billion cells down there, you can find a spontaneous mutation that will suppress the ACT1-1 allele. And the idea was to get at other genes that are regulating actin. And what Peter Novick, who was a postdoc in David Botstein's lab, found was SAC1-6 in combination with ACT1-1 allowed the cells to grow. And this is an example of genetic suppression. And we simply call that a positive genetic interaction. The double mutant grows better than either of the single mutants, or better than you'd expect. It doesn't have to grow better than either of the single mutants. It just grows better than you'd expect for the fitness associated with each of the single mutants. And the ACT1 should be dead, but it's alive here. Okay, so the wild thing was that when Peter Novick and David Botstein crossed ACT1-2, a different allele to SAC1-6, which suppresses ACT1-1, the double mutant was dead. And so there was a completely different phenotype, and they thought this was interesting. And this is what is known as synthetic lethality, and we call that a negative genetic interaction. And the way we think it's working is that ACT1-1 tends to prevent filament formation. SAC1-6 activates it. But you know that actin is dynamic. It has to go from a monomer to a filament in a very dynamic way. And if ACT1-2 stabilizes the filament, and then you combine it with this mutation that also promotes filament formation, then you can imagine that would lead to a synthetic lethality in this case, but a suppression in this case. Okay, so David evidently, he says that Peter was not interested in the double mutant that died, but David started doing some reading about it, and sure enough, there was a special name for two mutations, both of which are viable, that combine to create a lethal double mutant, and that's synthetic lethality. And this term was coined by a Drosophila geneticist, Dobzhansky, in 1946. And I think in the literature, fly geneticists, as early as the 20s, actually, there's this guy, I think it was Sturtevant, actually, it was, it's another guy. His name will come to me. But anyway, they were crossing some pretty sophisticated mutants around at that time. Amazingly, they didn't really even know what a gene was, but they recorded examples of this in the 20s. Dobzhansky coined the term in the 40s, in the mid 40s, and what he was doing was crossing individuals from outbred populations and looking for defects in development in the offspring. And he could trace the defect in development to a locus from one parent and combined with a locus from another parent. And so there definitely are mutations, natural variation in populations that when brought together can lead to an extreme double mutant phenotype like synthetic lethality. So just to go over that again, synthetic lethality is one of our favorite genetic interactions. It's an extreme negative genetic interaction. And basically it occurs when you delete one gene, the cells are viable, and none are viable, and they're viable, but the double mutant is lethal. And this is very rare. So, and that's why geneticists love it. There are, in yeast, which has 6,000 genes, there's 18 million gene pairs, 36 billion gene triplets. And so there's huge potential to take variation in a single locus, combine it with other variation in another locus, either as a gene pair or a gene triplet, and to create more extreme phenotypes. And we think that this probably underlies a lot of the genetics that we see within individuals and in any population. And so this is why we wanted to go around and do some systematic genetics and really understand what genetic networks look like. And it's fun because if you have, sorry about that, what am I doing here? If you have two non-essential pathways, A and B, each of which has 3 components, and if I can delete, say A1, and if I cross that to mutations in every single mutations in every other gene in the genome, and if there is a backup pathway like B, where a mutation here and a mutation here leads to synthetic lethality, I will find genetic interactions with all the components of the backup pathway, okay? That's the beauty of systematic functional analysis. And if you do that with another component of the pathway, you'll get the same results because it doesn't really matter if you kill the pathway at this level or this level. And so in the end, we get many different pairs that lead to the same synthetic lethality, and the pattern of genetic interactions is the same for genes in the same pathway. So we can cluster genes into pathways, and we can figure out which functional modules, whether a pathway or a complex, are talking to each other functionally through these genetic interactions. And that's the power of them. They don't necessarily mean there's any physical connection between the genes or their modules, but they definitely show you that there's functional connections. And ultimately, this is the type of wiring diagram we want for human cells so that we can understand how genetic combinations of genetic variation is going to affect cellular physiology and affect organismal function and diseases. So Lee Hartwell, who won the Nobel Prize in yeast for his cell cycle work, he developed a library of TS alleles in genes that were blocked at various stages of the cell cycle, and he put together a model for how eukaryotic cell division worked. And basically that was taking a very complicated process and dissecting it genetically at a time when most yeast geneticists were focused on simple biosynthetic pathways. So it really changed the landscape of the type of genetic approach that the yeast community was doing. And not only that, it homed in on all these, highly conserved cell cycle drivers. And but he's also very interested in just the general concept of synthetic lethality. And he articulated many years ago that you could use this for cancer therapy because cancer cells would carry cancer-specific mutations like, components of the DNA repair pathway that would lead to a mutagenic phenotype and then ultimately lead to cancer. And you could exploit those through synthetic lethality to develop therapies that would kill the cancer cells specifically. And so this was articulated, at the end of the late 90s. And he talked about looking for genes that are conserved from yeast to flies, figuring out what their synthetic lethal partners were, and then developing cancer therapies based on that if they were also found in human tumors. And now, of course, you can do this, very rapidly with CRISPR-like methods. And the DepMap project at the Broad is taking hundreds of different cancer cell lines and defining cell line specific vulnerabilities that are potential targets. But the other wild thing that he talked about was the potential for genetic interactions to drive the genotype-phenotype relationship. And basically, he was always impressed with the idea that you could have two almost benign mutations that on their own had no real fitness defect. But when you combine them, you get this incredible lethal combination through synthetic lethality. And his simple idea was that if this occurs in yeast, it's going to occur in all organisms, as we've seen with the fly genetics in the 40s. And that because there are so many potential combinations and so much variation in any genome, in particular the human genome, that genetic interactions will drive a lot of our inherited phenotypes. And this is an interesting concept because from the GWAS studies that people started doing once we sequenced the human genome, you could map common variation. And then we could score individuals for basically their haplotypes and all their common variation, and we could try and link that variation to various diseases. And what that revealed was, yes, sure, you could find some associations between common variants and diseases, but largely it also revealed this huge amount of missing heritability. And there's various explanations for that, including rare variants, additive effects of small effect variants. But the other possibility is that there are genetic interactions underlying that. And these have been difficult to reveal because it's a statistical nightmare. And one way you can think about that is that if you need a mutation if you have to sort of perturb the function of two different pathways because there's sort of backup systems built in for genetic robustness in eukaryotic cells and organisms, then there will be many different pairs of genes that could lead to a disease phenotype. And pathways are usually more complicated than this. So and here we have 9 different pairs of genes, but they're, if the pathway has 6 components, then there's many different pairs of genes that could lead to a disease phenotype. And then in cohorts of, several thousand people, you're just not going to be able to have any statistical power to find them. However, if we get huge cohorts, we could find a single pair underlying disease. And in this paper, Zook and Lander went through all those calculations and said that basically came to the conclusion that genetic interactions may be underlying a significant component of the missing heritability. I'm not going to talk about that today, but if you add prior information like, oh, these genes are in a functional module and these genes are in a functional module, we think that there are methods to look for connections between modules more than you'd expect by chance. So you gain, if you add prior information, you gain statistical power. And we're trying to adapt those systems to reveal genetic interactions from human genetic data. Okay, so let's go into yeast and apply SGA to the whole genome. And we do this simply by taking an array of mutants. They could be deletion mutants or TS alleles. We put them all out on a series of arrayed 1536 dishes, and then we simply cross in a mutation in another gene. And we go through meiosis. In a single colony here, after we germinate them with this special selection system we will have a pool of double mutants, single mutants, and wild type cells all in one colony. But because we mark the input alleles with 2 different drug resistant markers, we can select for the double mutant. And on the final plate, we have an array of double mutants. We start with an array of single mutants, cross in another gene. We end up with an array of double mutants, and then we score a colony size as a proxy for fitness. And you can actually move all kinds of things around like this. GFP alleles, we can make triple mutants. It's a fairly adaptable system. Amy Tong was a grad student. Amy Hinyang Tong, and she started the system, and then she applied it to several hundred genes as a grad student with a huge team of people working with her. But this is like a classic plate where we've taken this gene, BNI1. It's a formin that nucleates actin cables in the yeast cell that control polarized morphogenesis, okay? And it has a paralog, BNR1. There's the paralog, okay? And so the BNR1 double mutant has no formin in the cell. They can't make actin cables. They are big balls that grow isotropically and then die. And you can see these colonies are a little smaller than the ones around them, and that's what we quantify to score cell death in this pinning assay. And on this little plate here, you can see that BNI1 looks like it's synthetic lethal with BUD6 and CLOT4. And when we do tetrad analysis, and if you have two genes segregating in the cross, then 1 in 4 of our spores would be dead if it's a true synthetic lethal phenotype. And so BNI1 cross BNR1, we see 1 in 4 dying. In this particular tetrad, there are probably all single mutants, so nothing dies. But here we've got a couple double mutants, couple wild type spores. There's a double mutant. One of these guys would be a single mutant. Same with CLOT4. The BNI1 CLOT4 is dead. The BNI1 BUD6 is slow growing. And you know, in this plate, you can kind of see they look a little bigger, but it's, you know, so we can score synthetic sick and synthetic lethal negative genetic interactions by looking at smaller colony size. We can score positive genetic interactions by looking at larger colony sizes. And that's particularly easy if the query strain has a slow growth phenotype in the first place. So Amy did this for 8 different query genes. So this is one query. She crossed all 5,000 viables, and she gets these synthetic lethal and sick interactions. Here's another one, synthetic lethal sick. Here's one, synthetic lethal and sick. And the interactions here overlap with this other gene, and so on and so forth. And this, when we colored the genes based on their functional annotations, this made sense. So SGS1 is a helicase involved in DNA synthesis and repair, and it interacts with other genes involved in DNA synthesis and repair. So other pathways that when you compromise the cell by deleting this helicase, these other ones become very important. These two genes are components of the ARP2/3 complex, so they have similar functions in the cell, and that's why they share many genetic interactions in the cell. And again, they're involved in endocytosis and cell polarization, and they tend to interact with other genes involved in cell polarization. The actin cables that BNI1 makes are involved in polarized secretion, and so that's why we see these interactions with genes involved in polarized secretion. The actin cables are also involved in spindle orientation because there's a myosin motor that links to cytoplasmic microtubules and basically pulls the nucleus into the mother bud neck and orients the spindle while it does that. And the backup pathway, the non-essential backup pathway, is the Dynactin Pathway. And we see all the components of the Dynactin Pathway are lethal in combination with the deletion of BNI1. And those cells are alive because they've got, you know, this paralog here but they're not, the paralog isn't situated at the bud tip, it's situated at the bud neck. It's not a perfect replacement for this formin. And so that's why we can get these synthetic lethal combinations in these other pathways. And so even from our very first map, we could see that we were going to define functional relationships. And it looks like there's a lot of interactions. There are, like, so there's, you know, on the order of 100 interactions with BNI1 and, you know, maybe 50 with this guy or 25 or something. But remember, we're testing 5,000 viable deletion mutants. So it's still very rare that you see a genetic interaction. And then the ones you see are often functionally connected to the query gene. And so it starts to sort, the system sorts the parts of the cells into components that work together. And you can see the Dynactin pathway also interacts with this plus N microtubule binding protein. And so its role in spindle orientation is revealed through this interaction, and the role of BNI1 in setting up spindle orientation is also revealed through this set of pathway interactions. So that's the type of data, and we could use this then to argue that we should try and do this for the whole genome as a model, and potentially for doing it for human cells in the future. Even though CRISPR wasn't around, the idea was we'll find something like CRISPR where it'll work, and we'll show everybody how powerful it is. And so enter Michael Costanzo, a research associate here who spent the last 10 years really leading a team of technicians and working very closely with computational biologists to sort out this large scale project. We had a local engineer build us machines where we could load up on the order of 200 petri dishes, and on each dish that we could have 1,536 colonies, and we could score them for genetic interactions. Our computational team includes local heroes like Anastasia Berezhnikova, but we worked very closely with Chad Myers. First, he was a grad student in Olga Troyanskaya's lab at Princeton, and now when he's been leading his own lab. And he's had some very talented postdocs and graduate students. In particular, Ben and Carlos have played major roles in helping us figure out the signal from the noise and the properties associated with the signal. So when we do this, we take into account the fitness phenotypes of the single mutants and the double mutants. So if deletion of a non-essential gene lowers the fitness of the cell but doesn't kill it, we can measure that by the colony size. If we take a functionally unrelated gene that also has a fitness defect but doesn't kill the cell, we can record that. And when we combine this mutation in this background, we expect to see its phenotypes. We expect to lower the fitness of this mutant by 50%, just like we did a wild type cell. And that's our model for how the double mutant should behave. Of course, if the double mutant's dead, that deviates from the model, and we score that as a negative genetic interaction. If the double mutant grows better than we would expect, we score that as a positive genetic interaction. You could see there'd be various types of positive genetic interactions that you could score depending on the system. And we just go through all this. We do all kinds of batch corrections. We go from noisy stuff to some genetic interactions that we believe in. And this took quite a while to figure out, largely because of a batch effect, so that when we do screens on the same day, they'd often show lots of parent interactions in common. And so we would screen random genes that should not really show any interactions in common. We could normalize out patterns that basically were just batch effects associated with the robot, the technician, and their query array. And that really made a big difference, allowing us to get at true interactions. So when we do this with a gene, we score negative and positive interactions with a bunch of other genes on the order of 50. They're often functionally connected. So if we start with an actin gene, we see interactions with other genes that are involved in actin function. And we often see indirect positive interactions, where we activate a checkpoint and slow the cell cycle down, and therefore we don't see the phenotype of the mutant allele, or we disable the proteasome a little bit, and then we stabilize the mutant allele, so that often the positive interactions are non-specific. They're not these beautiful suppression interactions, and that's because we're crossing loss of function mutations to each other. We're not selecting for, suppressors like Peter Novick and David Botstein did. We're just crossing in loss of function mutations. So once you have a matrix like this, you can view it as a network as well. So that here I've taken the genes ABC in module 1, and I'm scoring negative interactions where you combine a mutation in one component of the module with another component of the module. So, and this often happens if the module is composed of essential genes. I'm crossing 2 TS alleles at the permissive temperature in the same module. 2 hits in the module will often kill the cell. And so you see negative interactions within the module, but we can also in this network format score what happens when we cross a mutation in gene A to a mutation in another module or another module. And in this case, ABC is negative interactions within, negative interactions with module 2, positive interactions with module 3. So you can view it as a matrix or a network. And the network is useful for showing all the data at once, but we have to tone it down a little bit. We can't show so many edges. So we like to do it in this format where the genes are nodes on the network. The edges represent their similar pattern of genetic interactions. So A, B, and C are connected by a short edge because they have similar patterns of genetic interactions, a very high Pearson correlation coefficient between those genetic interaction profiles. That's also true for this set of genes over here. And there is an overlap between these 2 modules, and they get a longer edge. And so on our global genetic interaction profile similarity map, that's this one, genes are connected to one another closely if they have similar patterns of genetic interactions. And genes within the same module have very similar patterns. Genes within modules that perform similar functions have similar patterns, but less similar patterns interaction. So you basically just sort all the genes in the cell based on their similarity of function. And we connect those with an edge that are highly similar in function. So we did this over 15 years, and then you get this global network. What Anastasia did, I'm just checking this, I don't have this here, but what she did was come up with an automated way of functionally annotating the network. So you can see these dense clusters. These are sets of genes that have similar function. And for example, all the DNA synthesis repair genes form one dense cluster. A bunch of genes in mRNA processing of ribosomal genes form a cluster, and they're these guys here and these guys here, and they're similar patterns of genetic interactions to the ribosome biogenesis genes because they're involved in processing those genes themselves. Genes involved in vesicle-mediated trafficking are clustering together. So this gets back to the idea of the model for the cell, and this is basically how we view the cell. We can see organelles and things in it, and Randy and other experts identified all the genes involved in secretion and their pathways, but this is fairly isolated. They're not connecting them to all the other functions in the cell because they're doing, intense specific biochemistry, cell biology, and genetics on particular genes. But the connections between those pathways and other pathways are inherent in this type of map. And one way to see that is by taking the map, taking the genes that show very strong profiles and looking at them in a hierarchical manner. So we can take them here. So genes in particular pathways all cluster together, and we can see that if we cut these dendrogram off at this level and look at specific modules, we can see the proteasome genes are clustering together. Genes involved in kinetochore function are clustering together. Genes involved in DNA synthesis are clustering together, or sister chromatid cohesion. If you go up another level, you take all the genes that have something to do with DNA synthesis and repair, and they form a bioprocess. And so that would be these larger clusters on the dendrogram. And if we go up at a higher level, we kind of get back to that model that was on the blackboard behind Randy Sheckman, where these great big clusters now correspond to compartments in the cell. So we have the nucleus, the nuclear compartment, the mitochondrial compartment, peroxisome, secretory pathway, nucleolus. And you know, what I like to point out at this point is that, you know, we have annotations for the genes. So SGD and the PhD experts that read papers and annotate gene function from very focused studies are important. And then we're doing this, taking that information and only our colony size information from single and double mutants, and we can put this functional map together for the cell. It's where we see that genetic interactions show pathways, complexes, processes, and compartments. Okay, so that's the double mutant thing. And we're trying to take that to human cells now. And what I was saying previously is we really need to define all the functional modules in human cells or any organism for that matter, and then how they're connected to one another by genetic interactions. And that will allow us to really start to interpret variation in an individual and how it's affecting the physiology of that individual. But I focused on digenic interactions, but there's more complex interactions. And Elena Kuzmin was a student in the lab. She started looking at trigenic interactions. Okay, so here it's the same game. If you take gene 1 and cross it to the array, you find out all the synthetic lethal double mutants and the suppressors. And you can do that for gene 2. You get all its synthetic lethal combinations and suppressors. But if these two are not lethal, you can make a double mutant, and then you can take that and cross it to the array and score for trigenic interactions, okay? And the issue here is there's always false positives and negatives associated with each one of these screens. And so in order to do a single trigenic analysis, we have to do at least 2. We have to do this screen twice, this screen twice, and this screen twice. So we have to do 6 screens in order to pull it off. And you can score trigenic interactions. In this case, you have to take into account single mutant fitness, double mutant fitness, double mutant genetic interactions, and then you can parse out the trigenic interactions. And the cool thing was we have a lot of quantitative measures that are associated with each gene on this network. So we could pick a gene, 2 genes that are in the same bioprocess, or a gene that's in this bioprocess and this bioprocess, genes that are very close on the network or further apart, genes that are highly connected on the network or are not connected that much. And you know, basically that's what we did. So we took double mutant queries that either had a low negative genetic interaction or a high negative genetic interaction, that were lowly connected or highly connected, that had sparse similarity of their profiles or highly similar profiles. And we scored for trigenic interactions. Here's an example where we did a double mutant screen with this gene. We get a bunch of secretion interactions. Another one with this, we get a bunch of secretion interactions. We could make the double mutant. We get more secretion interactions, but we also get interactions with genes involved in DNA repair. And we get an interaction with TOR1. And I'm not going to go into the details, but we think this double mutant basically gives us a TOR2-like phenotype, because TOR2 mutants show this type of pattern of genetic interactions. They're synthetic lethal with TOR1, and they have interactions with DNA synthesis and repair. So this double mutant kind of mimics a TOR2 and then expands our genetic interactions. Along here, you can see all the single and double mutant colony size, but we make the triple mutant, we get this impressive genetic interaction. So you can sort of see the general properties of trigenic interactions here. They tend to, the double mutant shows interactions with the same types of genes as the single mutants, but it expands into other functional categories. The trigenic interactions, like digenic interactions often occur with genes that have similar function in the cell, and so they're predictive and they define functionality of the cell, but they're often weaker and they show this tendency to show sort of longer distance functional connections. And but these properties, like the stronger the negative interaction with the two query genes, the higher their profile, the more functionally connected they are, and the larger their degree on the digenic network, that's highly predictive of which double mutant queries will show trigenic interactions. These trigenic interactions often modify a digenic interaction on the network. So the digenic network is predictive of trigenic interactions. Most of the interactions we see are modifications of a digenic interaction. However, we do score some novel trigenic interactions as well, and that's a substantial component of the network. In total, while there's 1,000 essential genes where a single hit will kill the cell, there's 500,000 digenic interactions that lead to a synthetic sick or lethal phenotype. And we can estimate that there's on the order of 100 million trigenic interactions. And so this really points to the idea that combinatorial genetics can lead to profound genotype to phenotype relationships. And in our paper, you could go into this, we really think that trigenic interactions themselves would probably drive speciation, so that if you separate two populations and they accumulate their own genetic or their own variation, you only need about 20 fixed substitutions before trigenic interactions will drive speciation, because none of the progeny will be viable. They'll always have a trigenic interaction that will kill them. Okay, so that study was kind of interesting because it's mirrored in this analysis of how conditions affect the global digenic network. And we just published this in May. And so in this case, what we're doing is we take query genes and we sampled the entire, genes from all over the network, genes of very different functions. And then we also use conditions that affect particular bioprocesses in the lab. So like tunicamycin, of course, blocks glycosylation. And so genes in this part of the map are hypersensitive to tunicamycin, and we've got query genes in there that we're gonna do digenic interaction screens in the condition of tunicamycin, and we walked over 14 different conditions. We only did 26 screens against a sort of a mini array of genes that behave well, but this is enough for us to sample how do conditions affect the global genetic interaction network. And when we just do it under normal conditions, we score G cross G interactions, but now we're gonna score G cross E interactions. Okay, so here's an example of some of the conditions we use, like Benamil. So if you take all the genes, this array of 1,200 genes that are all over the map, and we plate the single mutants on Benamil, we can score which mutants and which functions are hypersensitive to Benamil. So of course, genes involved in chromosome segregation are sensitive. Tubulin carries an intron, so genes involved in mRNA processing are sensitive. And the microtubule cytoskeleton affects vesicle secretion genes in yeast as well, because they're connected through this whole cell polarity thing. Monosin is a compound that affects vesicle trafficking, and most of the genes in this part of the network are hypersensitive to monosin. And we have query genes that are in these guys, but we also have query genes all over the place that we're scoring for genetic changes in genetic interactions under this condition. And, so this is the kind of thing we see where we have a reference set of interactions. In a condition, you might see a stronger negative genetic interaction. We're only scoring negative genetic interactions. You might see novel genetic interactions, and you might see some genetic interactions disappear. And so then in the end, we can use this difference between the condition and the reference to score a differential network. And you know, it's again, it's like the trigenic thing. You have to do every screen 6 times in order to get statistical data, and you score genetic interactions in the 2 conditions. Then you take the difference to score this differential network. It's fairly complicated, right? So imagine, you know, here's the different classes of differential negative interactions. So in the reference condition, if I have a positive interaction, and in the condition, if I have a negative interaction, and then I take the condition interaction, subtract that one, I get a stronger differential negative interaction. So it's gone from this to this is basically what we're scoring. If it could be a modified interaction where it's a weak, it's weakly negative in the reference, it's a little more negative in the condition, and so I still get a negative interaction, but it's not as strong as going from a positive to a negative. You go from a positive in the reference to a less positive, and when we do the subtraction, we score that as a negative. So this is the set of negative interactions. Perhaps the most interesting ones are the novel ones where we don't see any interaction in the reference. We see a strong negative in the condition, and the differential will be that. So this is the Benamel interactions we see. And there's these various classes. And, you know, we base this reverse situation. We don't think this is real. We score a few of them, but they're really weak, and they don't seem to be statistically sound. So there's not very many of those. But there are a number of the mass condition where we don't see a genetic interaction condition because the condition kind of mimics the interaction in the first place. And so we can basically quantify everything. And we do this under Benamel, but we also do it under 14 different conditions. And the bottom line is that most things don't change, okay? You know, at least, you know, something like 60 to 80% of the reference network is observed in the normal condition and in the new condition. A bunch of genes, we score them in the reference condition, but they show slightly different type of interaction or a degree of interaction in the reference condition. And we see a relatively small set of novel interactions. And if we do this over all 14 conditions, we get basically the same thing, where most of the genetic interactions we see in the reference don't change. Some of them are modified, and a small subset are novel. And this is very consistent. So these are the differential G by E's are rare, and the novels are even more rare. And this is kind of fun because there's been several papers where people have talked about massive genetic rewiring, and basically it's, when we look at their data, it's sleight of hand because if you pay attention to a lot of weak interactions, much of which is probably noise, then you can conclude that there's massive rewiring. But in fact, there isn't if you pay close attention to everything. And that makes sense to us because most of the genetic network that we see in the reference condition is driven by interactions with essential genes. And that's not going to change too much under a condition. So, so it's kind of cool. When we look at these novel interactions, they're interesting too. So G cross G interactions, as I've said, they occur amongst functionally related genes. So if I start with a query gene, I see interactions with other genes that have similar function in the cell, okay? The G by E interactions, they move out a little bit. So a bunch of them are in this next bioprocess, but we also see some of them over here in this bioprocess. And this is a general trend, so that the bottom line is that G by G interactions occur with genes that have similar function. G by E interactions often occur with totally distant genes, or slightly distant genes, and they're slightly weaker in magnitude. And so they do map new functions in the cell or in new functional connections, but they're tricky to tease apart compared to the digenic interaction map. Okay, so I'm guessing I should start wrapping this up, right, Yvonne?
Yes.
Yeah, so,
yes.
But I have to show you some, I'm gonna skip all this. Well, I can't skip it totally, but I can kind of skip it. That it's too much data for you to go through that. But the bottom line is we can do this in human cells now systematically, and we're expanding our collaboration with our team in Toronto to another researcher here at the Donnelly Center, Jason Moffat. And he's been instrumental in developing shRNAi screens and CRISPR screens. He did one of those 3 studies for looking at the essential genes in 2015, and we've a bunch of students that we're working together on. So the challenge is obvious: 6,000 genes, 20,000 genes, 1,000 essential, 2,000 essential, all kinds of double mutants in human cells, 200 million possible pairs just by going up, basically 3-fold in genes. And so we expect, tons of genetic interactions here. And we're trying to map these in a smart way. And what we've learned from yeast was that the essential genes have 5-fold more interactions than the non-essential genes, and that this is highly correlated with single mutant fitness defects. So if you're single mutant, whether it's essential or a non-essential gene, a deletion or a TS allele, if that single mutant query that you're crossing, or the gene on the array that you're crossing to, has a fitness defect, it tends to show a large number of negative and positive interactions. And there's other features associated with it, but that's the main driving feature. So we try and make query mutations that have a fitness defect in HAP1 cell line. HAP1 cells are haploid human cells. They have the core set of essential genes in them, which you can map, and they express about 10,000 genes. They're like any other cell line. The beauty of them, though, is that they're very efficient with CRISPR, and they don't have massive parts of the genome that are amplified, like HeLa cells, for example. So that if we do a genome-wide CRISPR screen with this, we can classify all the genes that are non-essential, those that have a sick phenotype, and those that are essential. And then we can pick those that are slightly sick, create a query strain with that, and then do a genome-wide screen in that background in order to score genetic interactions. And so basically that's what we do. We'll take, wild type, and we'll score how it behaves when we make a CRISPR mutation and a bunch of different genes. And we'll take a query, a congenic or isogenic query strain where we've deleted gene B, do the genome-wide screen, and then we can score synthetic lethal interactions in this background. And you do all this with barcode in a pool, with barcode sequencing as the readout. And just like in yeast, we can score negative and positive genetic interactions. And just like in yeast, this is a gene involved in metabolism. We score negative and positive interactions with other genes that often have roles in metabolism. And there's a nice paper in Nature Metabolism where we look at a few of these screens and correct those points. I was going to tell you about PTAR1 and its yeast ortholog. but it's cool. We see, if we take the yeast version of PTAR1, we basically get a map that looks like this. And the interactions are very similar. All the yeast snares interact with yeast PTAR1. And we even see suppressor interactions. In this case, PTAR1, if you delete it, the cells are sick, but you can delete non-essential genes and suppress that sickness. And we see the same types of suppressors in both yeast and humans. And so there definitely is an overlap between these networks. But of course, the human one is more complex. There's more pathways, so you see additional information. But yeah, there's the yeast screen. Here's the human screen. Here's the suppressors there. These are the same suppressors over here, basically. And we're starting to put together a network of functional connections based on CRISPR genetic interaction profiles, and we can see similar, functional enrichment amongst genes with similar profiles. So we're definitely opening the door to all the type of stuff we did in yeast in human cells. Of course, it's a lot trickier. There's less signal to noise, and it's a lot more expensive, but it will be done. And we can see nice clusters of genes that have similar roles, and we can identify new genes. So this is a new gene involved in lipid metabolism, and it shows similar profiles for other genes involved in lipid metabolism. Genes involved in DNA synthesis and repair, a new gene involved in DNA synthesis and repair. And so it's all coming together. And it's really just a matter now of making it happen. And eventually, once we make it happen, we think we can use the modules we get out of this network to look for genetic interactions associated with human genetics. And with that, I'd like to thank you for your attention. Here's everybody that's played a role in this work. We have 3 labs at U of T and Chad at the University of Minnesota, and lots of students and postdocs that have been working along the way. So thank you very much. And if you guys have any questions, I'd be happy to try and answer them.
Thank you so much for this talk. And those of you who have questions, si tienen preguntas, levanten la mano, las pueden hacer directamente o bien escribiendo. Entonces, Alex, tienes una pregunta, no sé dónde estás, pero vas.
Yo tampoco sé, ya estoy. Hi, Charlie. Hi, thanks for a very, very nice talk. I have a technical question regarding the use of temperature-sensitive alleles in your screens. Sure. So how do you rule out the possibility that some of the interactions have to do with the reversion of the temperature-sensitive mutation? Is that a possibility, namely that you would get apparently positive interactions because of reversion of the allele in all the process of the SGA process, namely?
Yeah, it doesn't happen very often. So it's true that, it wouldn't happen in the middle of a screen, for example. It could happen, we're moving them from freezer to freezer, and oh yeah, it gets a suppressor. And that does happen now and then. And it can also, especially if we have an array, we'll have a whole series of plates. They're all TS mutants, and we keep copying them. But what's really wild is that we find those when we do a cross, because now the fitness of that double mutant depends on the suppressor. And when you cross it to all queries over here, eventually you cross it to one that's right beside the suppressor. And you can't make the suppressor double mutant. And so when you select for the TS allele, which has a marker on it, and you select for your other gene, and if that other gene is right beside the suppressor, you can't get a crossover to get them in there. And so then you get a collinear set of what looks like synthetic lethal guys, and they map the suppressor for you. And so when we did the all by all analysis, we found out that, yeah, about 5% of the deletion mutants and 5% of the TS mutants we made, in fact, carried a suppressor. And then we would map it with this method, because we'd see it, we go, oh, there's like 20 guys all side by side who look really sick, all in a row. And then we could sequence that area and find the suppressor. And so the systematic thing really allowed us to annotate our whole set of arrays and figure out which ones had suppressors and which ones didn't. And so I guess surprisingly, it's only a small subset that do. But it was really useful, because then we started, going after suppressors, right?
That's nice. Yeah, something you were not after and you finally get. And one other quick question. So your maps do not include genes having to do with the sexual cycle, right? Because of the way you get the mutants. Is anyone trying to look into that? Would there be a way of looking to genes having to do with mating and sporulation?
Yeah, no, you have a good point. If you're just involved in mating, you can't even, if you're required for mating, we can't score genetic interactions with you. So then we'd have to, well, there's always ways, right? Like you could make TS alleles of those genes. You could do some fancy stuff. And then the sporulation thing, you have to get double mutants in a diploid. There are things to do, Alex, that we have not done that must be done. That's what you're pointing out. And it would be really nice to do it for sporulation, because it's a very complex developmental program. And on our map, there are, of the 6,000 genes, there remain about 2,000 that we can't put on the map, because when we do digenic or trigenics, they show nothing. And I suspect a bunch of those are involved in sporulation. And it would really be nice to crack that set of, like we call it the dark net. But yeah, I think we'd have to go to these other developmental programs in order to figure it out. And so it's a really good point. And someone should do that in Mexico, right?
Cool.
Yeah, it could be a national project.
Yeah, thanks, Charlie. It was nice seeing you. Bye.
There is one question in the chat, so maybe you want to read it rather than me reading it. It's from Elsa.
Oh,
there's several questions here in the chat. One from Elsa, and then from Emiliano and Eli.
A couple of years ago, it was proposed to use Yeast's method for bioremediation due to their ability to adapt to extreme conditions. Although metabolic response is well studied, it's known how gene interactions change and how epistasis is involved in order to cope with temperature stress. Is it known how gene interactions change or epistasis involved in order to cope with temperature stress or heavy metal environments? Yeah, so that would be very much like the condition stuff that I briefly went over. I mean, I think you would want to take all the single mutants, expose them to these stress conditions, and then look at genetic interactions under those particular conditions for genes that you think are important for that function. And you're going to find some. Most of the network's not going to change, as I said, but there'll be 7% of the interactions you find that will be novel. Some of those could be critical for adapting to that condition. And so it could definitely be powerful. A lot of we found when we went over 14 different conditions that it was a subset of genes that appeared to participate in these novel interactions. And I think that the reason we found a subset of genes was because many of the conditions create a stress physiology state in the cell that's similar. So it's possible that our study would point to which, some of the genes that would participate in that. But yeah, that seems like if you really want an answer under a particular condition, then you do the condition type analysis that we did under that particular condition. Does that make sense?
Mm-hmm.
Okay. Then anyone could, say something. But should I go on to the next question?
Yeah, I guess so, because they're all going this way.
Okay. Great talk. Thank you very much. We know that the CTCFs in humans form topologically associated domains TAD in chromosomes by acting as insulators. So I was wondering if there are CTCFs analogous genes in yeast, and if you can detect TADs by analyzing the positive gene-gene interactions. I don't know anything about CTCFs or TADs. So I'm afraid that I can't comment on that. Does anyone else know if CTCFs?
CTCF is not present in yeast.
Okay.
It's not there.
That's why.
That takes care of it.
It doesn't, but yeah, I mean, in human cells, you should be able to do that, right? I mean, that's the beauty of moving to another model system. And then, this type of genetic interaction can be done on any function that you're interested in any model cell or organism. And so I think it's a good idea to attack something like that that's important. And then if it's not in yeast, you just go do it in the appropriate model, right? Okay, to what extent do genetic interaction networks built with double mutants resemble those done with, for example, mRNA expression or protein level, since both networks provide information regarding functional protein? What additional information can the former networks provide that might not be obtained with other systems and biological approaches? Yeah, so this is I think, an important question. Like there's lots of groups doing large scale things, and we should be able to integrate that data in a way to make the most accurate model of the cell, right? And we're trying to do that with the yeast data that's available. And there are some issues though, like with the genetic networks, and to a certain extent with the gene expression networks, you get a nice profile that you can use easily to create networks and to integrate them in with machine learning or AI systems. Protein-protein interaction networks, you don't often really get a profile. And so that's harder to deal with. And yet the information, the link between one protein and another protein, if it is done in a low throughput way, that's a very, information rich linkage, but it's hard to put an A-B link into something where we have these nice profiles and then weight it properly in order to make sense of it. The other thing I think that are issues with integrating data is that the data sets are very sparse. So like the protein, we made a huge effort to do an all by all analysis of genetic interactions in yeast. Of course, it's not all by all, we say it is, but it's a pretty solid analysis. In comparison, I think the protein-protein interaction networks are full of false negatives and false positives, depending on how they've been done. And so in the end, you end up with a network that actually, apart from these stable complexes like the proteasome, the ribosome, our knowledge of protein-protein interactions, especially dynamic ones, is very, very poor for yeast and especially for human cells. And so there actually isn't very much information associated with protein-protein interaction networks, even though we all intuitively value them and understand what they mean. And then the gene expression profiles, that data is good for certain functions, you know, like ergosterol biosynthesis in yeast is regulated at the transcriptional level, and those genes all show similar profiles when you perturb them, but that's rare. And often these profiles have hundreds of genes in them. And so there is no functional connection between the query gene and the response genes. It's possible that, you know, the single cell analysis and different tissues and more complex organisms and you know, intense transcriptional profiling will create data sets that will be useful. But if you try and, you know, in yeast, look at the three major types of functional genomics data set: gene expression, PPI, and genetic interactions. It's not only is it hard to integrate them, but it's hard. There isn't a lot of data associated with the gene expression data, and the PPI data is full of false negatives. So I think there's a lot of work to be done there, but eventually we should get to a point where we can combine this data and pick and choose which parts of it that we use for an integrated network that's better than any single network. Okay.
Then we have Anel's question, I think.
Yeah. Could these gene interaction networks be used to infer gene annotation from the biological pathway they are members of? Yes, for sure. And the only problem there is some, like SGD has curators, PhD level curators that use Go annotations to annotate gene function, and they purposely don't use high throughput data to do that so that we can use those annotations to assess the quality of our high throughput data. And if you start using the high throughput data, then there's circularity in, you know, whether your data is reasonable or not. And so they can be used to define function, but we have to make sure we know when and where we're using that information and how we're using it. YouTube, very impressive work. Looking ahead, could you comment on the possibility of predictive power of these findings, and related, any ideas on how to evaluate the role of noise in the networks? Yeah, so we often, we try to assess the predictive power all the time, and it turns out that the stronger the signal between a genetic interaction pair of genes, the stronger that negative or positive signal, the more predictive the power is, and that it's more likely that those two genes are annotated to the same function. So you can, if you have a quantitative analysis, you can cut your profiles off at various levels, the strength of your interactions off at various levels, and you can look at the predictive power of linking two genes that are annotated similarly through either their profile or their negative interactions. And we've done that in our papers. You can see that. And the noise in the network, yeah, I mean, I think that's a really good point. That one of the major issues is distinguishing signal from noise. And when you do these large scale things, there's a lot of noise associated with it. And again, hidden in the fine print of the supplementary material, we have a false negative and a false positive rate. And we get those by doing things like screening with gene A, getting a profile, and then if we take an interaction, if A interacts with B, we screen with gene B and see if we get this, the gene A interaction. You can play that game over and over again. The other thing you can do is just do the same screen many, many times, right? And figure out what's the noise and what's the signal for a particular screen. And then you have to look at your experimental approach and somehow model the noise as well. And so there's various methods to do that, none of which are really that satisfying because we constantly struggle with signal to noise ratio. The simplest thing though is that the stronger the signal, the more likely it's real, and doing so that often seems to hold true. And doing repeats of each screen definitely gives you an idea of how much noise you've got, and then you just have to hone your experimental technique to try and drive down the noise. But I think there should be computational methods to try and weight what we think is not noise, and then somehow learn the properties of those interactions within a sea of noise at the same time. But we haven't really solved that problem by any means. And so it's something we think about every second of the day, basically. You nailed that one. What kind of genetic analysis bring more information to create the genetic interaction networks? Could these change with different models like yeast, cancer, tumor cells? What kind of genetic analysis bring more information and to create the genetic interaction networks? Yeah, I'm not quite sure what you mean by the first phrase or first question. Could these change different models? So do you mean, whoever this is, do they mean like single mutants and double mutants? I'm not sure what kind of genetic analysis brings more information. Maybe someone could help me out there. Maybe Mario could say, or Yvonne.
I don't know either.
Well, I'm not sure. I mean, probably other genetic analysis could be overexpression.
Oh, okay.
Gene or something like that is what I imagine, but I'm not sure.
Yeah. So, yeah, there's definitely you can do your query genetic perturbation could be a single loss of function mutation in a non-essential gene, a TS allele mutation in an essential gene. It could be a double mutant. It could be, if you overexpress a gene, you perturb the stoichiometry of things, or you activate a pathway if that leads to a gain of function type phenotype. So all of these genetic perturbations can be used to score genetic interactions. That's a good point. And in yeast, we found that mutations that lead to a fitness defect are the best queries. And it doesn't really matter what it is, whether it's overexpression, deletion of a non-essential gene, or a TS allele of an essential gene. As long as you have a fitness defect, and this is true with chemical genetic analysis too, the compounds that slow the cell down, they're the ones that give us genetic interactions. And so in any model system, I think that's going to hold true. It holds true in yeast and human cells. The functional connections between the genes in our hands are the strongest just for plain digenic interactions, combining two loss of function mutations. If we do what we call synthetic dosage lethality, we turn on a gene, overexpress it, that slows the growth down, we'll get a bunch of interactions that are reproducible, strong, and we can score them, but they often don't really have a functional connection to the query gene. I mean, they might if you overproduce a gene and it has something to do with, high levels of that protein are toxic to the cell, and then you delete a gene that's involved in the turnover of that protein, it'll show a strong genetic interaction, and you'll find it, so that's cool. But you'll also find all kinds of other things that you really can't make any sense of in our hands. And so, yeah, I think with any system, you just got to, survey some of those things and see what's going to work. And it might be the opposite for tumor cells, for all I know. But I would survey a few things and check it out, and then go to town on what's delivering.
I don't know. So, Yvonne, you have off your microphone. Okay.
Yes, I'm sorry, but so I guess we're finishing, and thank you so much for sharing all of that. It's a humongous amount of work.
Well, thank you for inviting me, because it's hard to find a captive audience that will listen to me for an hour. Really appreciate it. Yeah.
Okay.
And
thank you very much.
Thank you so much, and we just hope that you were here.
Okay, in the future, I'll pop down.
In the future, maybe next time.
In the future, yes. Okay, that's a deal. Thank you so much for sharing your time with us too, and that's it then for the next time.
Okay, nice to meet you. Bye.
Nice to meet you. Thank you.